Okay, thanks everybody. If you could take your seats, we'll start the second part of the livestock session. So I would like to invite Tina Sørensen Dalgaard from Aarhus University to speak to us about avian coronaviruses. And Tina is one of our keynote speakers. So thank you for joining us.
Thank you so much, and good morning, everyone. Thank you very much for the opportunity to come here today to present to this very varied audience. I'm very excited about this. I must say that this is an area that we have not been working with for a very long time. So indeed, some of the results I will show you today are quite preliminary, so I hope you'll bear with that. And I also hope it will still support my take-home message that it could be worthwhile to look into the species-specific biology to sort of understand how we can target different adjuvants to different species. We are shifting from alpha and beta coronaviruses to gamma coronavirus. You will appreciate that we still have the four structural proteins, and also in the chickens we have variants, a lot of variants with differences in the S1 subunit of the spike protein. So it's not that different really. The virus causing disease in chickens is called infectious bronchitis virus, and it is airborne and highly contagious, and we are talking about respiratory disease. What we see is actually not, it's quite a mild infection. We do not see a lot of mortality, but we do see predisposition to other viral and bacterial diseases that may cause mortality. And what is particularly noticed in layers is this malformation of the eggs and also a decrease in egg production. So it is actually a huge economical problem in the poultry industry. So it is infecting and replicating in the upper respiratory tract, and it deciliates the cilia on the epithelial cells, and it can spread to other parts of the body. We also have some particular nephropathogenic strains that cause lesions in the kidneys, and we do have some local strains, but also some strains that circulate globally. And it is actually a remarkable amount of different variants that we see, despite the fact that we, or maybe because of the fact that we for decades have been using live attenuated vaccines to try and control the disease. We have commercially available live attenuated vaccines and also some inactivated vaccines. But the problem is that they confer protection towards the strain that is actually included in the vaccine. I was particularly happy about the question earlier this morning about variation. And this is something we see a lot in chickens. Infections and vaccinations, there is a lot of difference depending on the genetic background of the animals. And for decades, people have been trying to come up with clever breeding selection strategies in order to breed chickens for disease resistance. And what we do know is that chicken MHC locus, the B locus, is highly affecting whether or not an animal is disease resistant or susceptible. And we've had a lot of fun trying to understand this. And over here you see a review on different MHC haplotypes and which ones are indeed naturally resistant to infectious bronchitis virus and which ones are highly susceptible. So that's one thing causing variation, and obviously not only in the natural infection, very much also in the vaccine responses that we see in chickens. This is from an old study where we've used some of the university lines. We've got some inbred lines with highly characterized MHC haplotypes, and you see that the humoral and the cellular responses to a live attenuated coronavirus vaccine is really varying between these inbred lines, but also very much between individuals within a line. And these particular studies, there has been a lot of them, were like in an experimental setting where each individual had a particular volume, a particular dose of the vaccine. And this is not at all what happens out in production, where very often spray vaccines are used, and you sort of also count on a spread between the flock, or within the flock. So the individual vaccine take is very variable, which makes things very complicated. So what we do know about protective immunity against the avian coronavirus is that maternal antibodies, they are somewhat important, but already after 1 week after hatch, they are not really that protective anymore. We do know that humoral immunity is important. We do know that if we remove the bursa fabricius, the B-cell organ in chickens, they actually experience more severe illness. We have had a lot of data on the specific antibodies in serum, and it doesn't really correlate that well with the clinical signs. But we do know that local mucosal antibodies, neutralizing antibodies, are obviously really important. Something that we're really interested in are the T-cell responses, and there are some old studies with adoptive transfer of cytotoxic T-cells, and for sure they are very important in the protection. We are not so fortunate in the chicken community. We do not have any tetramers available, so it makes our life a little harder when we assess the T-cell mediated immune responses in our world. But what has been done, there is no easy way of doing it, but when we try to assess if a vaccine is protective against the heterologous strain of virus, it is done by these protector typing experiments, where you vaccinate with one strain and you challenge with another, and it's really, really laborious. You need to use SPF chickens, you need to challenge, you need to have endpoint measurements, and it is actually a lot of work to come up with the protector type matrix. This is an example of such a study where you prime with one strain and boost with another, and maybe challenge with a third strain. And then we come up with some recommendations that if you combine these two strains, you may or may not get cross-protection against a third strain. So complicated and tedious. We also have a problem with long-lived immunity. In Denmark, actually, the layers in the production period are currently vaccinated every 6 weeks. It is actually a huge problem, and the quality of the vaccines are not that good. And it may not be a sustainable strategy to keep trying to figure out these protector types. What we've been looking a little bit into is subunit vaccines, and in theory that seems quite attractive. You can maybe combine different variants of the S protein, and for sure there is not the risk of reversion to virulence as we see in the live attenuated vaccines. But there are also some prerequisites that needs to be met, and ideally we want vaccines for spray or drinking water. Because you can imagine if you have 10,000 chickens, it is doable in the hatchery, but in the production period, it's really no fun to inject vaccines into this many animals. So what is needed is really to figure out clever adjuvants for these types of vaccines. We have been particularly focused on making sure that T-cell response would be induced. And what you see here is like the classical T-cell activation dogma, where you have an antigen-presenting cell, and you have some MHC molecules, and you activate CD4 and CD8 T-cells. And the secreted cytokine sort of determines the flavor of the T-cell response. That's very well described in mammals, and we know it happens in secondary lymphoid organs. But when we come to the chickens, we do have some peculiarities in the chicken immune system. For instance, chickens do not have normal organized draining lymph nodes. When we talk about respiratory viruses, they do have head-associated lymphoid tissue. They've got the Herderian gland behind the eyes. So it's not so that we do not have any tissue to study the interactions in, but the model systems are slightly different. Luckily, the antigen-presenting cells are somewhat similar. We have good data on chicken dendritic cells and macrophages, so we have cell model systems that we can use to sort of assess some of these mechanisms. And what we've been working a little bit with is targeting antigen-presenting cells. By this example shows a TLR ligand, the TLR receptor on the surface of the antigen-presenting cells. You couple it with an antigen, and hopefully that makes sure that the antigen is internalized and presented on MHC molecules on the dendritic cell. And in theory, that's a really neat system. And you can pick and choose. There are so many nice pattern recognition receptors that you can actually maybe target with your adjuvants. And what we've been focusing on are the C-type lectin receptors. It is not trivial, though. The C-type lectin receptors recognizes carbohydrates, glycans, nice, cheap compounds, biodegradable, sustainable. But depending on what type of glycan you choose and what receptor you target, you get different responses. Some are actually inhibitory, so you need to pay attention to the combination of glycans or the particular glycan or the particular receptor that you want to target with your adjuvant. We have been looking into the many genes encoding C-type lectins, and it is known from mammals that this is a huge family, and they're divided into different subgroups, and some are even soluble. But what we do know for the membrane-bound C-type lectin receptors is that they are important in viral infections, although there are also examples of virus exploitation where a virus could use the receptor to gain entry into the host cells. We have been focusing on the membrane-bound receptors in group 2, 5, and 6 as they're expressed on myeloid cells. And we started by looking into the literature and found some nice chicken-specific receptors, and we thought, yeah, that looks lovely, except that some of them are, you recognize some of them from the mammalian literature, but some of them are really avian-specific. Interestingly enough, some of them are located in the MHC complex on chromosome 16. But we started out by asking ourselves, is this all there is? Are there any more genes that we could actually go for? So we did some data mining and some in silico identification by blasting the carbohydrate recognition domain of one of the known genes. And in total, we only identified 28 potential candidates, and 10 of them were actually previously described. We did identify novel ones, some very avian-specific, and also some highly expressed in lymphoid tissue. So we made a little table of candidates, and we looked for putative sugar and calcium-specific binding motifs, and we looked for endocytosis motifs, and we very tediously looked at bone marrow-derived dendritic cells and macrophages to see if the receptors were even expressed in the cell type that we wanted to target. And in this work, again, we saw huge impact on MHC haplotype. These receptors are expressed to varying degree depending on the genetic background of the chicken, and that adds to the complexity, really. And the functional relevance, that's really the important part of this. One thing is to identify a bunch of genes, but then what? And we've got sort of little pilot studies going in different directions, as there are, some of them do not have any true mammalian orthologs. There is no jumping the fence, really. We have to do the work, and we have to figure out from scratch what is happening. So we are building fusion proteins and doing glycan arrays to figure out the ligands of the receptors. And there are also some machine learning-based software that you can use to predict the glycan ligands and so on. So that's one pilot project. And then we've also been doing a little bit of in vitro screening and in vivo studies. And I will show you one of the examples of one of these pilot studies in the later slides. This example is a pilot study where we identified a ligand by in vitro studies. We took bone marrow-derived dendritic cells from the chicken. We cultured them with a bunch of known mammalian C-type lectin receptor ligands. And then we looked a little bit about activation status of the cells and endocytosis of different targets, and also the possibility of proper T-cell activation. You are not probably able to see the details here, but this slide is about activation of the dendritic cells, and we actually had a candidate called pustulin that came up in numerous studies, and we also looked at the cytokine profile and comparing it to what happened when we cultured the cells with the natural virus. We also looked at endocytosis, and we decided that this pustulin, that was actually a really interesting candidate for an adjuvant approach. We did try with recombinant N protein. It's not as diverse as the spike protein, as you know. So we did produce some recombinant IBV-N proteins, and we pulsed MHC-matched bone marrow-derived dendritic cells with this recombinant protein plus minus pustulin, and we were actually able to show that pustulin made a huge difference in terms of activating at least CD4 T cells. So in parallel, we actually also did the in vivo studies, and interestingly enough, we had a little bit of difficulty in buying enough pustulin to do a proper chicken experiment. It is actually derived from a fungus, from a ligand-forming fungus growing on cliffs exposed to strong winds. But we got our hands on a bucket of pustulin, and we set out to do this animal experiment. But we collaborated with the National Vaccine Institute in Denmark that had some nice liposome platform that we could sort of tap into and add the pustulin on top of that. The liposomes that we used were actually also containing a mingle ligand and a TLR3 ligand. So it was shooting on a bunch of receptors to actually facilitate a good outcome. This is the experimental setup. We had like some control groups, and then we had the liposomes without the pustulin and with pustulin, and then we compared to the live attenuated vaccine. And to our huge disappointment, only the live attenuated vaccine were actually able to reduce the viral load upon challenge. But to our slight excitement, we did see some tendencies when we looked at the amount or the frequency of activated T cells in peripheral blood upon challenge. And actually this pustulin containing vaccine also showed us lower amounts of activated T cells upon the challenge. So that was at least a little bit comforting for us. And also when we, by flow cytometry, looked into cytokine production of the IBV-specific T cells in these chickens, the pustulin vaccine were actually also able to induce IL-17 and interferon gamma responses comparable to the live attenuated vaccine. So by all means, there are a number of knowledge gaps. We simply mean to suggest that it is possibly worthwhile to understand a little bit about the biology when you sort of engage in these types of endeavors. For certain, we have established that there are not so many genes, not so many receptors to target in the chickens, and it might limit the exploitation of this strategy. But at the same time, the chicken genome has been really poorly annotated, so we're still hoping that we might have some missing genes that we have not yet identified. For instance, the chicken TNF-alpha gene was discovered 6, 7, 8 years ago. We had a really hard time finding it, so you see the annotation of the genome is really not incredible. But more work is definitely to be done in identifying the ligands and also making the correct formulations if this is going to be successful. So thank you to a number of people who have helped in these preliminary studies, and thank you for letting me share the presentation here.
Thank you, Tina. Are there any questions from the audience? Louise?
Thanks very much, Tina. Very interesting talk. I was going to ask about the mucosal delivery in these adjuvants, because is that going to be difficult that you'll get a suitable adjuvant to work with that kind of delivery system? I mean, what about pustulin, for instance? Do you know that that works given by that route?
Say that again?
Do you know if the pustulin works if you would give the vaccine mucosally? I'm not sure what route you vaccinated the
Yes,
chicken.
I'm sorry, I didn't say that. This was actually intranasally, so it was a mucosal administration of the pustulin. That's the whole purpose of trying to develop new subunit vaccines. Yeah. So yes.
Okay, thanks very much.
Okay, any other questions? Oh, Dylan, you're not allowed to escape yet.
I'm going back.
So if you're using sort of innate ligands to try and prime a response, I wonder, does that in any way inhibit live attenuated vaccines? You know, if you perhaps triggering an antiviral state before the virus has got a chance to sort of replicate and make its antigen?
Yes. I actually took out a few slides in the interest of time, but we also have these soluble C-type lectin receptors, and in the very old days, we tried to just take live attenuated vaccine and add a little bit of glycan of various kinds, chitosan, phospholigosaccharides, stuff like that. And we weren't quite sure what to expect, if that would sort of, it wasn't covalently linked to the antigen in any way. So we weren't quite sure what would happen, if it would inhibit sort of attachment of the virus, or if it would go via these soluble receptors. And what we did see in these very old studies was actually a huge increase in the humoral responses to live attenuated vaccines when we combined it with sugar, just in solution. And we don't have any data on T-cell specific immunity in these very old studies. Yeah.
Thank you for your nice talk. My name is Zana Mahmoud. I'm from Liverpool University as honorary associate. So in the proteomic point of view, have you looked in the glycosylation of your targeted, genes or regarding the pointing the receptors toward IBV, because you know IBV structural glycoprotein they are heavily glycosylated.
Exactly.
Thank you.
Yes. No, that's obviously an area of research too, to try to mimic the glycosylation of the virus and sort of to direct the immune response in the right way by mimicking this. Yeah.
I've got one last question before we move on to our next presentation. So, with the modes of delivery that we need to do for chickens, I guess we've also got then potentially innate immune reactivity when you're actually dosing these birds. And I know that that's also quite a difficult area. Have you been investigating the innate response when you load these vaccines in as well?
No, we have sampled a lot of samples, but that is also studies to come.
Okay, great. Thanks very much. I'll hand over to Louise to do the introduction for the next one.
Okay, thanks very much. We have our first offered paper of the session. Pleased to invite Olivier Touzelet to come and speak. I know Olivier is not going to talk about animal coronaviruses, but repurposing drugs for SARS-2.
Yes, thank you. Yes, thank you for the organizers to invite us to give a bit of data on our ongoing project. So this is an MRC-funded project that we're leading here at, we're undertaking here at Queen's with collaborators in Oxford and Liverpool. And the aim of this project is to repurpose drugs, approved drugs, to use them as combinations that may have antiviral activity against us, so that we could use to treat COVID patients. So the advantage of combinations, there are two main advantages of combinations over monotherapies. The first one being that they offer synergy. So we could possibly target multiple steps of the virus cycle, entry or boarding or replication. And one other important aspect of combinations is the may allow to overcome resistance, which is obviously a common feature of many viruses. And it's particularly true in the context of SARS-CoV-2. There's already variants have been identified carrying mutations that are giving some level of resistance to clinical protease inhibitors, such as nirmatrelvir, which is one potent inhibitor that's been used in the clinic. And we also know for SARS-CoV-2 that a combination of monoclonal antibodies will definitely limit the emergence of resistant virus if you compare to a single treatment with a monoclonal antibody. So what I will try to do in this talk is to take you through the process of how we start with a large library of large number of drugs and how we filter it down to interesting hits that may be transposable to humans, and I will focus today mainly on double combinations. So in terms of the screening itself, so we are using two cell lines for the screening, Calves, which are human epithelial cell line, which is a cell of reference to study SARS-CoV-2, and VATs, which are viral overexpressing human ACE2 and TMPRSS2, which are the two main important elements for the drive entry of SARS-CoV-2. And we start with three main drug libraries. So we designed what we call a customized library, so whereby we cherry-picked drugs from the literature. We used a low-pack library, which is a library of small pharmacology active compounds, and a library of FDA-approved drugs. In terms of screening itself, the cells are incubated with the drugs for 24 hours. Then they are infected, adenomyv01. We are using, for the screening, Delta variant. And 3 days later, we're measuring the CPE using a fluorescent assay. And so the first part of this project was to what we call do a primary screen, which is whereby we just tested the activity of the single drugs of each of the libraries in order for us to sort of establish a threshold of drugs that we can use that have the most likely chance of being successful as a combination. So this is just the raw data from this screening. So this is the percentage of CP inhibition across the 2 cell lines, across the 3 libraries. And I'm just putting you just a list of single drugs that we have identified in orange here, showing at least 80% inhibition across the 2 cell lines for each of the libraries, just to show that we did, so we use a couple of positive controls here. So remdesivir, which is known to have anti-SARS-2 activity, casirivimab, which is a neutralizing monoclonal antibody, to show that it did show a strong CP inhibition, so that was a way to confirm that the robustness of the assay. And we did also recover some known drugs, such as imaltravir or molnupiravir, which have already been approved into the clinic. So that allowed us to select the best drugs to use for the combination screening. And for this screening, we use what we call a music approach. So this approach consists in clustering the drugs in groups of 5, when it's randomized in a way so that all the drugs are eventually paired with all of the other drugs of the library. And that really allowed us to reduce the resources needed for this combination, as opposed to if we're using a simple pairwise combination. And for the rest of this talk, I will mainly focus on the customized drug library, because it's the most advanced library in terms of screening. And because it had a low number of drugs, we included all these drugs into the combination screen. So in terms of the data that we covered, so this is the initial screening, combination screen, using the music approach. So this is the CP inhibition in y-axis across the 2 cell line. Each dot represents a group of 5 drugs. And when we repeated this assay multiple times, we could identify across the 2 cell lines 103 groups of 5 drugs that showed over 90% CP inhibition. Now at this point, what we do then is we deconvolute. So what it means is out of all the 303 groups of 5 drugs that we identified, we then test again all possible pairs that may be working together in the screen. So from this, we repeated this screening again with the pairs, and we identified across the 2 cell line 46 pairs that showed over 90% inhibition for the 2 cell lines. So obviously that's a lot of combination to test. So one thing we want to do is we wanted to rank them, and rank them based on potency. So we established dose effect curves for all of these combinations. So each graph is a percentage of CP inhibition against the drug dilution of the combination. And for each combination, we tested in black is the dose effect of the combination against the single drugs that made that combination. That allowed us to rank all the combinations based on potency using IC50s and IC90s. And it also allowed us, importantly, to look at and select the most synergistic combination, meaning the combinations that showed an increased potency when they are combined compared to the potency of the single drugs alone. And we use IC50s and IC90s to measure this. And from that list of 46, we could filter it down to 27 combinations of interest. Still a high number. So what we did then is we tried to rank them based on clinical profiling. So we used 3 criteria for this. We used the clinical stage of the drugs that were included in those combinations. We used the route of administration, and we looked at the safety profile, the known safety profile of those drugs. And so we created this sort of a color-coded system to rank all drugs that we had. So we had red, orange, yellow, and red, depending on the stages of these on these criteria. And when we apply this to the 27 combination, so you can see some of the combinations appeared red and orange, so they were eliminated or not prioritized, and so on. And using a combination of potency, synergism, clinical profiling, we thought we had isolated 9 combinations of pairs of drugs that showed good potency, good synergism, and looked to be seemed okay using our clinical profiling. Amongst those combinations, some of the drugs that were involved are already known activities against SARS-CoV-2. They were protease inhibitors of the 3CL or TMPRSS2. Some of the drugs here had suspected mode of action against SARS-CoV-2. Some of the drugs for which there are no reported information as to how they would work against SARS-CoV-2. So obviously, this was all done using the high throughput method. So we were obviously interested in validating these using more robust virology assessment. So what we did in this case is we used A549 cells, or AAT cells. So they are A549 overexpressing human ACE2 and TMPRSS2. So these cells were incubated with the drugs for 2 hours before to be infected at an MOI of 0.5. The virus was removed, and the cells were further incubated with the drugs for 72 hours. So as from before, we worked out dose effect curves for those 9 combinations. Again, this is the percentage of virus inhibition against the drug concentrations. In black is the inhibition curve of the combination against, in red and blue, the inhibition curves of the single drugs that made those combinations. And really the important point here to remember is that we isolated 5 combinations that looked very promising. Combination 3, 4, 5, 6, and 7 looked to be the most potent and most synergistic after virology validation. It was all very promising. And the last part was the pharmacometrics analysis, which to me was very novel. So I'm going to try to explain to you what we did. So we wanted to work out which combination was the most likely to be transposable to humans using the in vitro data that we had and some of the clinical data that were already available. So because we have to take into account for the drugs the pharmacokinetics and the pharmacodynamics, how the drugs are absorbed, metabolized, excreted in people. And really, this is all inferred from clinical trials. And one value that we're interested in is what it's called the Cmax, which is the highest amount of drug that is available at any time point in a person. And what we were interested to know, and what we were told that was important to know, is that the IC90 of our combinations in vitro would have to be lower than the Cmax, and the lowest, the better. The lower, the better. So what we did then, for the 9 combinations that we had isolated, first off, there's only 3 combinations for which we could work out the pharmacometric profiling, because the Cmax data wasn't available for all of the drugs that made those combinations. And out of the 3, which are 4, 5, and 6, combination 5 is the one that stood out from the rest, because it showed that the Cmax of each of the drugs that made that combination exceeded the IC90 of our combination by more than 60 folds and 127 folds. So out of all the screening that we've done, so far combination 5 is the lead combination based on all the available data, so clinical data and in vitro data. So I hope I've made it clear how we proceeded to select that hit, and we're still doing it because we're still screening more drugs. And I will leave you with this update. So this is a multi-layered project between us and our partners. So we've completed stage 1 here, which is the HTS, the high throughput screening. And we're now navigating between stage 2, 3, and 4, depending on the library we're talking about. And with the hits we've isolated, we hope we can move into stage 5 and 6 very soon, which involved an in vivo model, which will be done in Liverpool. And as well, we want to try using therapeutic model, using alley cultures to test those combinations. So I would like to thank all the persons involved in this project, our collaborators in Oxford and Liverpool, the team at Queen's, particularly Arflem, who's been working on this with me since day one, who is really the brains behind the project. So thank you for your attention.
Thanks very much, Olivier. Are there any questions for Olivier's talk? Yeah.
Thank you for your talk. I've actually got a couple of questions, if that's okay. So I noticed you always added the drug before infecting. Are you going to add the drug either during the infection or post-infection?
So all through the screening, the drugs were added before infection because we wanted to find, we wanted to be in the best conditions for the drug to work. Now that we've isolated promising combinations, what we're trying to do is one of the stage 5 of the project here, where we're going to use a therapeutic model in alley cultures, so which is a more advanced respiratory model, and we're going to do this a therapeutic approach where we're going to probably treat during infection, after the infection, and test how the combinations perform in that setup.
Okay, thanks. And my other question makes sense. My other question was, are you going to passage the virus through cells in the presence of the drugs to see if they evolve any resistance?
Yes, so that's this part of the project will be done in Oxford. So they are all set up to start this. So hopefully as well, this is something will be starting imminently because we have hits for them. So.
Cool. Thanks so much.
You're welcome.
Sorry, just a very quick question.
Yeah.
Thanks for the talk. If you administer a drug like, or a combination of drugs like this, say, I'm guessing like intranasally, is that the idea, or is it?
So at the minute, all the drugs we're selecting are orally, have an oral delivery. So this is one of the criteria we're trying to select, either oral or nasal.
And so if you administer intranasally, then how long would a drug like that hang around in the upper respiratory tract? Do you know?
So this would be probably for the drugs that we're trying to select. We're trying to select drugs for which there are clinical data available, and that type of information would be already known. So it's very variable. Obviously, in our assay, the drugs seem to be pretty efficient in the context of our assay, but obviously in persons it would be much different. But that is probably, I haven't looked personally, but that data is already available for the combinations we're selecting. That's one of the criteria where we wanted to be able to go into humans as quickly as we can. So we need to sort of target it to the best drugs that may be usable in humans. So that's, yeah.
Okay, thanks very much, Olivier. We'll move on to the last talk.
Thanks very much. So the last talk of the livestock coronavirus session is from Hannah Turkington from AFBI, and she's going to talk to us about bovine respiratory disease, coronaviruses in calves. Thank you.
Excuse me. Thank you for the introduction. So the work I'll be presenting today is collaborative, so I wanted to first acknowledge a couple of people. So first Rachel Bell, a former postdoc with us, and also Caitlin Hull, because they presented a lot of the data that I'll be presenting today. So what we are interested in is understanding bovine respiratory disease complex. This is one of the most economically important diseases for the agri-food industry. It is the main cause of cattle mortality from the age of 1 month to 2 years, and it costs the cattle industry around $1 billion every year. Generally, it's understood to be caused by an initial virus infection, of which there are a number of BRD-associated viruses, and then this predisposes calves to secondary bacterial infections and therefore more severe disease. So we can do 2 types of studies. So we can look at experimentally infected animals or animals that are naturally infected with BRD-associated viruses. In terms of experimental infections, we have two viruses that we mainly look at, so either bovine respiratory syncytial virus or bovine herpes virus. These studies generally last only for around a week before the animals are euthanized, so we use them to study early immune responses to these viruses. And we can also do metagenomic studies to identify genetic markers associated with susceptibility or resistance to infection in these calves, and this could be used to inform future breeding programs. In terms of natural virus infection, we had a case around 2 years ago where we had 40 calves brought into our animal services unit, and a lot of them developed pneumonia. At the time that they developed pneumonia, we couldn't sample them because they weren't under license. So what we did was later on, when we sampled them, we tried to retrospectively determine what the cause of pneumonia was. So we looked at the humoral and cell-mediated immune response to various viruses. And through this, we also were interested then in understanding the longevity of the maternal antibody response in these calves. And so these studies enable us to gain knowledge to inform the development of appropriate vaccination and treatment regimes for calves, with an aim to reduce the use of antibiotics and therefore reduce potential antimicrobial resistance. So here's an example of a challenge model. This is for bovine RSV. In the case of this virus, it's administered via nebulization. And then in the days post-infection, we can measure a number of different parameters. So we can look at the rectal temperature, the respiratory rate of the animals. We can measure clinical signs of disease. We can look at the virus shedding from the nasal by taking nasal swabs and doing qPCR. We can look at the gross lung pathology at post-mortem, so identify any lesions or signs of disease. And we can also look at virus replication in the lungs with immunofluorescence. In terms of bovine herpes virus, it's similar, but for this virus, we have to administer it via atomization. So this is something that we test beforehand, which delivery method is the best for whatever virus we're using. And again, take our various parameters over the length of the study. In this case, this study only lasted for 6 days, but we already saw signs of disease post-mortem in the nasal turbinates of the animals. So with our two different types of studies, we can take a lot of different samples from the animals and then use them in further assays. We can look at specific antibody levels against the viruses. So we can look at IgG and IgM levels in the blood. And we can also look at mucosal IgA from the nasal secretions and use ELISAs to look at antibody levels. We can isolate peripheral blood mononuclear cells, or PBMCs, from the blood, and we can use ELISpot and MTT assays to look at proliferation of the T cells as a marker of their activation. And as I said, we can also collect tissues from animals at post-mortem. We can collect lesions from the lungs and also compare them with nearby healthy tissues and isolate bronchial lymph nodes. And we've had previous collaborations with our colleagues in Chagas in the south and University of Missouri, where we've done transcriptomic analysis on tissues isolated from infected animals compared to uninfected animals, and these results have been published, and I won't be going into particular detail there, but I'll just show some data we have from, in this case, a bovine herpes virus challenge, where we looked at PBMC proliferation and cytokine production post-challenge. So these animals were euthanized at day 6 post-infection. As expected, we don't see a specific antibody response at this time point. However, we did already see a suppression of T cell proliferation measured by the metabolic activity being reduced in the T cells 6 days after infection. And we also saw a reduction in the cell's ability to be, so these are cells stimulated with bovine herpes virus antigen. There's a reduction in interferon gamma secretion, IL-17A, and IL-2. So those were just 3 cytokines we chose to look at there. In terms of our natural infection study, so just as a reminder, we had 40 calves that came on site where many developed pneumonia in the autumn of 2021. We didn't take any samples at this point, but when they were around 3 months old in January, we took blood samples, and then 4 months later in May, when they were around 7 months old, we did a further blood sampling, and we also, at that time point, got some nasal mucus samples. So we wanted to look at specific antibody levels against a variety of viruses. So in this case, it's for bovine RSV. We looked at the levels of IgM in the sera. This is the January bleed and the antibody titers of the 40 individual calves. So in January compared to May, we see a reduction in the number of IgM positive animals. And for IgG levels, we looked again in January and in May. Again, a bit of a reduction at the later time point in IgG positive animals. And we also looked at neutralizing antibody levels, and in this case we saw little or no neutralizing antibody response. For bovine coronavirus, again IgM in January and in May, and IgG in January and in May. So we see an IgM response, but we don't see any animals are positive for, or little positivity for IgG and for bovine coronavirus. And again, no neutralizing antibodies were detected at either time point. For bovine parainfluenza virus, again, IgM in January and in May, and IgG in January and in May. Most of the animals were remaining positive for IgM and IgG at the later time point. And in this case, we did actually see some animals with some neutralizing antibody response against bovine parainfluenza virus at both time points. For mucosal IgA antibodies, so this was only looked at the 7-month time point. This was the only time point that we took nasal mucus samples. And again, we looked at our 4 viruses of interest, and we see for the majority of animals, it seems that there's higher IgA present for bovine coronavirus and bovine parainfluenza virus than for RSV and herpes virus. We wanted to look at some cell-mediated immunity, so with Ellie spots, again looking at production of cytokines from PBMCs that have been stimulated with inactivated virus. So again, our 4 viruses, and we see in general there's a range of high and low level responders, but no significant difference between the viruses. So this would be a measure of T cell activation against these 4 viruses. For IL-2, again, a range of high and low level responders, but we saw a bit more response against bovine parainfluenza virus. So to summarize, in our experimental challenges, we've seen with transcriptomic analysis that a week after infection, we have a high number of differentially regulated genes and microRNAs with enriched KEGG pathways in infected animals compared to uninfected animals. And this is even the case when we didn't have a large clinical signs of disease. So there was still a good difference between infected and uninfected. In the case of bovine herpes virus infection, we see an early suppression of T cell responses. In our natural infection study, so given the age of the animals, we assume that what we're measuring are maternal antibodies, and that these calves have maternal antibodies against the different viruses that we tested, and that they are decaying by 7 months. However, most of the animals remain positive or had positive antibodies to bovine parainfluenza virus at the later time point. And so from this, we assume that this was an active immune response, and we therefore presume that bovine parainfluenza virus was the most likely cause of the pneumonia in the calves, also based on their neutralizing antibody levels, the IL-2 responses, and as I said, the longevity of their IgM and IgG responses. We did see a lower level of IgG specific for coronavirus compared to the other viruses we tested, and conversely, higher levels of mucosal IgA for coronavirus than for BRSV and bovine herpes virus. So these studies have increased our understanding of the early immune response in the calves, the longevity of the maternal antibody response, and possible markers of active as opposed to passive immunity. We don't quite understand why there is a difference between bovine coronavirus and the other viruses we tested in terms of their antibody or maternal antibody levels. So this would require some further investigation. So I just wanted to, again, acknowledge everyone who assisted on the projects.
Thanks very much, Hannah. Are there any questions from the audience? Thank you.
So why do you think there's so much maternal IgM present? And were you surprised that there's more IgM than IgG in these animals significantly past their clinical disease?
Yes, I think we were somewhat surprised. You would expect it to be more IgG than IgM. All I can say is that IgM is passed maternally as well. And yeah, the ELISAs aren't necessarily, it's more of a positive negative, so we can't maybe read too much into the levels, but that could be something that could be looked more into, what exactly the difference in the levels is.
Okay, thank you. Are there any others? I've got one question, and I'll do that while I'm walking up there, here. In terms of the bovine respiratory disease complex for your field calves, so it looks like the pneumonia was caused by one particular virus, because potentially.
Yeah.
But I'm kind of interested in the whole idea that actually there might be multiple infections going on in these calves in those early stages after the maternal antibody starts to wane. So, exploring the virome or the pathobiome, which might be viruses as well as bacterial infections. Yeah. So are you looking at field cases to be able to sample them while they're clinical, as well as looking at the immune responses?
Certainly, collaborators do more of the field sampling. We would be more of the experimental challenge aspect of it, where I guess we do focus on one specific virus and then see after infection how that impacts like the microbiome of the calves. But yeah, it's sort of a minefield, because there are so many different viruses and bacteria that are associated with it, and trying to understand which might play a more important role, or is it really a combination, or does it, this overall umbrella of BRDC, it can be in different calves caused by different diseases, and therefore which vaccines you should be using to try and prevent it. And at what time points do you vaccinate? So yeah, it's hard to unravel, I guess.
Similarly for the pigs, I guess, in terms of porcine respiratory disease complex. So are there any other questions for Hannah? Cyril?
Well, since you bring up this issue of the differences between all these viruses, I've always been curious. You may not have the answer to this, but since you bring it up, you didn't mention BVD. And
No.
I remember that, in the UK, you think, IBR is the predominant respiratory virus for bovine. If you go to continental Europe and France, it's BRSV. Do you have any clue for these differences in terms of the prevalence of these different respiratory viruses in the different cattle segments, in the UK and Europe?
I guess it just really does depend what's circulating. We did. I can't remember. Did we
So what about BVD?
look at BVD? They didn't present with any diarrhea. But yeah, maybe Louise can answer better.
We did test them for BVD, but they were negative. But then we have a BVD eradication program, which is quite successful. So there's probably less of it around. But it might be another place.
So I guess the other question is, what is the question that you're asking about these clinical animals when you send your sample in for testing? And quite often the vets will ask for one pathogen. So we test it for one pathogen. We don't all have the resources then to go, well, if that pathogen is negative, what is the differential diagnostics? But maybe, the power of next-gen sequencing and multiplex PCRs is going to be able to give us the ability to start to ask those questions and start to understand the complexes in virome versus pathobiome, rather than just asking, is it this particular disease or another one, actually having an understanding of the spectrum that's involved. So, there were lots of questions and lots of answers, so you need to keep all of those in mind for tomorrow's workshops, where we'll be talking about gap analysis, all about coronaviruses for livestock, wildlife and companion animals. We want to collate what those gaps are and how we might, as a research community, address those for the future. So keep thinking about those for tomorrow afternoon. I'm going to hand over to Louise now, who's got a couple of announcements before lunch.
Okay, I just wanted to say something. You'll notice out there beside the registration desk, we also have a stand for the Royal Society of Biology. I'd like people to take a look if they're not members. I'm a trustee for the Society and also on the local Northern Ireland committee here, and it is a great society to join at any stage. There are different levels of membership, ending up with fellow, which is a very recognized kind of status qualification, as are the memberships. There also can be certified as a certified biologist. There are lots of there are travel grants and things for early career researchers, etc., and lots of different meetings organised. And it's not just, although it's based in the UK, we have an international membership. So it is, please take a look at the stand, and there's literature there to have a look at. And I think a few free pencils and things. So yeah, we're going for lunch now. So we, during the lunch, I think we have an hour and a quarter. So please take the opportunity to look at posters as well.